Verónica Dumit is currently working at the General Federal Institute for Risk Assessment, and also is an expert in proteomics. So my question right now is, what is risk assessment? I have no idea. I don't know how proteomics could make any unveiling of the risk assessment. So Vero, many thanks for coming, and the stage is yours.
Thank you, Nico. Yes, first of all, thank you, the organizers, very much for the invitation. I'm really happy to be here. And well, it's also a good opportunity to present what I am doing now. I'm not working anymore at the university. I'm doing something different, but I still work in the field of proteomics, and I'm trying to bring proteomics into a new field. And as you can see here, the title of the talk, it's quite ambitious, and it will take a while until we get there. But let's start, first of all, with myself. You can see that I studied biotechnology at the university in Rosario, and then I continued with a PhD, also at the university here in Rosario. And during this time, I got a fellowship to study in Germany for a bit. At the beginning, 3 months, it was a DAD fellowship, and it worked well, so we could get a second fellowship, and I spent 2 years in Germany within the PhD, but yeah, I got the title from the university here. And when I was finished, I wanted to continue studying in Germany. I liked it very much. And this was the time I found a position at Jörn's group. He offered me a position, although I had no experience at all in proteomics. And that was the first time I could learn. So it was not new for me, proteomics, but I had no experience. And it was that time I had 3 years postdoc. And then I ran a core facility as the head of a core facility for proteomics, also in the university in Freiburg. And well, then, as you can see here, oops, sorry. There is a break, that it's the maternity leave. And after that, I moved to Berlin. And I am now working in an institute that does risk assessment. It means it evaluate everything that you consume, everything that you can buy, if it's safe for your health or not, for the consumer's health. And I worked in the department, as you can see here, for chemical and product safety. And the unit, the group where I am, it's in fiber and nano toxicology. So we work with nanomaterials. Because it's a long talk, and I don't know how long I will talk today, or until when, and what I'm going to continue tomorrow, I want to give you now the take-home message that for me it's important. So it is that omics will help to reduce, at least to replace or to reduce animal testing. And that I find wonderful. And your contribution is needed for that. Everyone doing proteomics or omics, can be transcriptomics, metabolomics, it's needed for that. And these are the different topics that I will cover during the talk. And I start with risk assessment. Nico wants to know what is risk assessment. So maybe, well, this is the institute. It is in Berlin, and it is located in 3 different parts. I work here, that there are labs and there are offices, and there are different fields of competence. So it's a food safety, food and feed, so what the cows or the animals eat, that it's important to know if it's safe, the food that is produced. Also biological safety is important, experimental toxicology we do there, and the safety of products, if the toys that you have for your children, they are safe, if they have pigments or if they have something that can be harmful. This is what we do at the institute, try to assess the risk of that. It's also pesticides, fertilizers and all that. It's also important for the institute, and we do risk assessment of that. And very important is the risk communication, because it has to be something that it's open to the public. And if you, as consumer, have any question if something can be safe, you can always contact the institute. You have a form for that. And this concern of you will go to any of us, the people working at the institute. The last one I got of these questions from the public, from the consumers, was about the masks. If the fibers in the mask can represent a harm for us, because we are breathing that constantly, all the time now, everywhere in the world, so can that represent a risk for the health? And well, I researched a little bit what is the equivalent of this in Argentina, and I could not find one institute that does exactly the same. But the UNMAT, that you probably heard about, especially now with the vaccines, or SENASA. I also found the CONAL that I did not know before. And the equivalent in the USA would be the FDA, that it's the Food and Drug Administration, but the institute where I work, they don't deal with drugs, so pharmaceuticals are not covered. Well, I can continue a little bit about the institute. So we belong to one of the ministries in Germany, Food and Agriculture Ministry, and what we do, it's independent. There is a budget that you can see, it's not bad, it's over 100 million a year, and the idea is that we can provide one opinion on the risk of the products, but that it's completely independent. And the staff, it's over 1,000 people, and half of them are scientists. And yeah, so there are many activities, and most of them, they have to do with communication. How do you explain people what is safe and what can be a risk? For example, if you cook and something is burned, this burned things can also represent risk. That's all part of the things that we do and how we try to communicate. And well, the importance, as for example, a question that always comes from the public is about sunscreen. Sunscreen, it's recommended everywhere, but still many of us think that it's or could be maybe toxic or potentially toxic. And it's even worse now that we know that there are nanomaterials in this cream. So everyone is concerned about the nanomaterials and what is it with the sunscreen. Do you think it's safe? Do you use it? Do you avoid it? And if you don't know, you can go to Google, you can look for that, and you will always find whatever you want. People saying, I won't stop using it, but some other that say, nanoparticles in sunscreen, they cause Alzheimer. And if you continue a bit further, you can even find some articles, research articles in decent journals with a good impact factor, where they show, and actually if you read, they show that the titanium dioxide particles could cause some central nervous system impairment. And if you read it, it's convincing. But what my institute does, and what we tried also to explain people, it's the details of that. What does it mean for humans? In this paper, they work with mice, and they found out that if they give this dose to the mice by inhaling, so the mice have to put all this amount through the nose, then they will have some impairment in the nervous system. They will behave weird. And it's everything significantly proven, so the research is good. But what would it mean for consumers? So if you make the equivalence, if you extrapolate the amount to humans, it will be the equivalent of inhaling this amount of titanium dioxide through your nose, which, it's not the same as a sunscreen. So, and this is the way that we can show to the public if there's a risk or not. For risk assessment, it's important the hazard, and here the hazard of these nanoparticles is clear. So the nanoparticles could cause Alzheimer or one of these impairment. That's the hazard, and that is clear. But it's also important what is the exposure and how it's the exposure. Is it a dermal? Is it an inhalation? It's something that we eat. That makes the difference. And also the dose, how much of that we use. Because the dose is what makes the poison. Yeah? So if you consume a little bit, nothing will happen. It's just a high dose that will cause the effect. And altogether, this is the risk characterization. So the end of the evaluation. And as you see here, hazard and risk are not the same. And this it's important for us. For example, a shark, the hazard is clear. The shark could kill you, and no doubt. But as long as you are not in the sea or at the sea, well, the risk is zero. Yeah? And what we do, it's a toxicology. We are always doing toxicology at the institute. This is what it's important for us. And what I want to make clear is that there are two different branches. You have the regulatory toxicology, that is what we do at the institute, but also basic science toxicology. And there, in basic science toxicology, you do whatever you want. This basic research, you use the techniques you want, and for example, the paper I showed before could fit this criterion. You do your research as you want, of course. But for regulatory toxicology, you cannot just do whatever you think it's right. Everything has to follow the regulations, and all the experiments that you do in this has to be exactly as it is expected. So you know which is the organism you will know, you know how many animals you need, for how long, how many doses, and so on. It's all very well regulated, and when you want to do regulatory toxicology, you have to follow these rules. And what I also would like to explain is that in the institute, we don't do this risk assessment. We don't do the experiments ourselves. If you are a company and you want to sell your products in Europe, you have to do the risk assessment yourself. You have to prove that what you produce or what you import is safe. So the company is the responsible for doing the experiments, showing the risk of them. And what the institute does, it's just to check that everything is correct, that the experiment followed the regulations, and that what the company claims as result, it's also what it should be. So, well, toxicology, you probably know, you probably heard what it is about. And for toxicology, in this regulatory one, what is important, it's the endpoints, the endpoint. Everyone cares about the endpoint. All the experiments are like that, endpoint experiments. You have a substance, and you have the guideline, how you have to proceed. And the only thing you care is what happened at the end. So you had the substance, you gave it to mice, and then you see if the mice have tumors, or if the mice have some problem in the skin. You just care for the effect. About the mechanism, for regulatory toxicology, we don't care. We just care for this end effect, for this end point. And there are many different categories of things that you can check, depending what you are selling or you are importing. And you see, it's endless. There are many more, but it's always like that. You make the experiment and you see what happened at the end. But how that happened, we don't really care. And most of these endpoints for most of the substance, they involve in vivo tests. And the animals that you need, the amount of animals and how long it takes and how expensive it is really unbelievable. And also Europe is trying to go further from animal testing, also the USA. Here's an example how a guideline, that is how you do the regulatory experiments, how it should be for neurotoxicity here. I don't expect you to read, but it's clear that it has to be in rat. It has to be by oral administration. It has to be at least 20 animals, 10 females and 10 male, at least 3 doses and a control group, and you have the strict times, how long it should take, and then what you have to measure. And for carcinogenicity, when you want to see if a substance can cause cancer, the experiment in mice, it takes 2 years. If you have to do it in rat, in addition, it takes 3 years, just for one substance. And the cost of how many animals and how long they have to be exposed, it's really, really very, very high. There are also some other concerns. So do you have this interspecies, can you extrapolate results from rat or from mouse to humans? Yeah, sometimes it's useful, but it's not always the case. There are also the ethical aspects and the time and the costs that it represents to make such experiment. Now I want to go to the second topic, that it will be about alternative methods. Do we have any, or which ones we could have? And these 3 R principles that were mentioned there, it is about reduce, refine, or replace animal tests. But now, if you want to do that, if you want to have alternative tests, you cannot have these endpoint experiments anymore. Then you will need to know the mechanism. So if we really want to find some alternatives for animal tests, we will have to go to the mechanism of the substance. And that's why proteomics, so this is not only my opinion, but it will be the future of replacing animal testing. Proteomics and omics techniques in general, they will be needed, and the efforts started already in this direction. So when you want to develop some alternative tests, it's not that you just develop one and it will be used. You are always welcome to propose some alternative test, but the relevance has also to be checked, and how this will represent really the toxicity of the substance. And the way it works in Europe, it's that when you come up with some alternative test, you can hand it in as a country to the OECD. Probably you've heard about the OECD. It's the Organization for Economic Cooperation and Development. It's very important. Many decisions are taken there. And this involves 38 countries, and most, Europe and Canada, the USA are part there. And all of them, all the member states, revise everything that has been submitted as alternative. And all of them, all the countries will comment, will show their concerns, and the guideline that you propose will be adapted, changed, or well, it will change along the years. And such a process takes many years. It takes a decade if everything works fine, because you have these iterations of, okay, we have a guideline, we have to comment, we have to adapt, we have to resubmit, it's much worse than with a paper. Okay? But the guideline that it's approved and that it's accepted by all the member states, then it will be used and enforced by all the countries. Meaning, if you want to do the risk assessment, all Europe, USA, Canada, and all the countries there, they will use that for the risk assessment for all the substances. So it's not a minor detail that you can come across with something like this. If you can bring proteomics there, if you can bring your assay and you can have it run, it's, well, it will be important. Not in the scientific way as making a paper, but for policies, for when the substances in the market, it's really relevant. Here, there are many guidelines that are out there that use in vitro tests. All of them, they are independent of animal testing, so you see that things are moving. So now there are many in vitro guidelines, but still, none of them is good for nanomaterials. That nanomaterial, it's what I work on, so that's none of them it's good for that. And none of them involves omics yet. No, that's, but it will come. For example, in skin sensitization, I'm sure one of you at least had an allergy, skin allergy, because of jewelry or a piercing, tattoos, or deodorants, or some perfume, or cosmetics. That it's very easy to develop an allergy. And before, all these tests were done in animals. But now, for cosmetics at least, it's not allowed anymore. For cosmetic purposes, you are not allowed to use animal trials to see if the substance is safe. But there are alternatives. And the reason why it's been accepted like this, it's because we know a bit about the mechanism of that. So you know that the substance has to penetrate the skin. It has to be bound to some proteins that are there. This will lead to the activation of the dendritic cells. There will be a migration to the lymph nodes, and then these lymph nodes will go back and they will secrete some cytokines or some interleukins and so on. So because we know the mechanism, how it is, now, okay, this was the experiment as it used to be before, when you had to do it with animals. As you see, you had the substance, you have to treat the mice for some days, and then apply some radioactive thing that will mark the dendritic cells, and then the mice are killed, and the lymph nodes are extracted and checked for radioactivity there. And this was done for everything that they wanted to check if there was could be some skin allergy. And you see, this is the minimum that you need for one substance. So minimum 15 animals, it takes 1 week, and still you see the performance. So it's 72% of predictivity, that the assay will tell you exactly what it's for humans, meaning it's not great. And still a lot of work, very expensive, and involves animals. So now, the mechanism I showed before, it's represented into the AOPs. This is important for regulatory purposes, for toxicology, for what we do, that you tried to describe the mechanism into key events. So as I mentioned, the key events, it's that the substance penetrates the skin. It's another key event, it's that it binds to proteins, and then that the dendritic cells are induced and are activated, and then there's a secretion of interleukin. These are all key events. And inflammation is another key event that you have when you have allergy. And now, if you try to explain the mechanism like that, and you have the different key events, what it's been developed, it's test for each one of these key events. Of course, with one of them, if you see, well, can my substance activate some dendritic cells, that's not enough to say, okay, this substance will cause allergy. It's not enough. But if you have a series of these guidelines, you will be able to know if the substance then can cause allergy. So now, if we go with in vitro guidelines, we will need several of them to know what will be the adverse outcome, what will happen, what will be the effect. But this is also when you go to the doctor and you want to know if you have, I don't know, an ulcer or cancer or something. It's never one test that they do. Maybe they start with a blood test and then they recommend, I don't know, X-ray or something, and it will be like that. And then they will confirm that you have some disease because, well, it was proven with different assays. And in this case, it's kind of the same. And what, for me, this AOP and skin allergy, it's very interesting. It's because there is one assay that's called the GARD that involves transcriptomics, and this is the first one that can tell you if the substance will cause allergy. So to check this key event, the number 3, you can use a transcriptomic test. And this is not yet completely approved, but it will be soon by the OECD. And here, it's a simple assay. You have your substance and you have the cells, and sensitizers, meaning the ones causing allergy, they will activate the cells, and those that are non-sensitizers not. And what they did, they had many substances that they knew they are allergenic and some that are not, and they did transcriptomic with this simple setup, and they checked for 29,000 genes, and they found out that 200 of them can be considered as the fingerprint. So if you check 200 out of them, they will give you a result if the substance can be allergenic or not. And then they perform these microarrays only on these 200 genes. And the predictivity, it's much better than animal tests. So this is performance, it's really, really much higher. I think it's very promising, and it's not yet approved, but it will come. It will be soon. But still, it's not proteomics. Nanomaterials, as I mentioned, I care for them because it's with my group, it's involved with them. And a nanomaterial, here you have the definition. The definition for institutes like mine, it's really very important, and it refers to a material with 1 or more external dimensions in the size range between 1 and 100. And here I'm showing different nanomaterials for carbon. They are all carbon allotropes, and they have great potential. But there are, of course, many more. You have titanium dioxide. It's a nanomaterial present in sunscreen. There are clothes also that have nanomaterials so that they are impermeable, and some plastics with nanomaterials to make them more resistant. Nanomaterials are being everywhere. But we still don't know if there could be some effect for health. And what you see, this is one of them, but graphene. Graphene, maybe you heard about it, is so interesting. You have, it's a flat, it's a sheet, only a monolayer of carbon atoms, but each carbon atom, it's bound to 3 of them. It means it has still one orbital that can transfer electrons like nothing else. And as conductors, they are wonderful. And if you mix a little bit of them to plastic, the plastic will be a conductor. And it transmits electricity like a cable, or even better. You can have the same, this flat sheet, all wrapped up like a carpet, like here, and this is a multi-wall carbon tube. And these are fibers that make, if you put them to other materials, it makes them very resistant, and also they can transmit electricity. So the nanomaterials, the potential, it's really, really amazing. And considering that this could be a solution for electric car batteries, we could imagine that this will be present everywhere very soon. The potential, it's really amazing. But what is the concern with them? So we had the example of asbestos. I'm pretty sure you know about them. I used to call them amianto when I was a child, and at school we even had something like this. We still have they are completely forbidden. They cause health problems. And yeah, they were great for isolation. These are fibers or silicates, and they can isolate like really well, and no other material. They were used in constructions, they were used in the trains, and everywhere, really everywhere. And everything was fine until 25 years after the asbestos started appearing. Some people that had worked with asbestos developed mesothelioma, and this is a lung cancer that is very aggressive. And yeah, the cases were really many. In Europe, there were very many in the USA. And although the mechanism is not yet completely clear, it is well understood that these fibers, they stay in the lung. And apparently they are inert, they don't do much, they stay there. But still the immune system tries to clear them out, and macrophages tried to attack them, try to phagocyte them, but they cannot because they are so long and rigid. So the macrophage will come to the fiber, try to clear it, and will die. And this will lead to more macrophages coming and generating oxidative stress, trying to get rid of all that, but dying while doing so. This will attract more macrophages, and so on. And you have this long latency, but that will develop into a cancer that is very, very aggressive. And actually, in principle, carbon tubes are the same. The substance, it's different, but the morphology, it's the same. And here, this toxicity, it's morphology related. And that's why the question is, what could it be if we use carbon tubes, and in many years we found out that they were very toxic. And this is one of the researchers projects I have, and the one that I enjoy the most. And it's how to assess frustrated phagocytosis, so this process in an in vitro assay. How can you do that? I still don't know how, but I work on that. Nanomaterials I've seen, they are also important in agriculture in Argentina. Sorry, this is in Spanish, but I took it from the Aprecid magazine. And you know Hugo Permichat, and he works at the university. And here in this, it's a 3-page long interview to him, and he explains all the benefits of nanomaterials when you use them as a carrier. So you have like a ball, and inside you can put fertilizer, so that the fertilizer can come out not all at the same time, but I don't know, along months or years, and that it's a very good fertilizer. Or you can have herbicides with the same, or, well, it's endless. But these are being used, although he even mentions that the evaluation of the risks, so this risk assessment, well, it should be, somehow should be done. And yeah, I showed this slide before to show all these guidelines that are accepted for in vitro, not in vivo, but none of them are used for nanomaterials. And that's why I tried to develop one for frustrated phagocytosis in vitro. Oops. And here I care more about inhalation because, well, I mentioned before this mesothelioma, it's a cancer in the lungs. And yeah, with the nanomaterials, it's the concern. If people working with them, they could inhale that, and what could happen in the lungs. And that's why, for me, lung, it's central in my research. And these nanomaterials, potentially, they could cause fibrosis or lung cancer or some other adverse outcomes. And all the tests to challenge this, to know if this could happen, they are in vitro. So that's why fibrosis and lung are, for me, or lung fibrosis, so important when coming to nanomaterials. And the way that fibrosis in the lung, it's understood, can be represented like that. I showed the same way of thinking for the skin allergy, and yeah, it's like this, key events. It's if a substance gives fibrosis, it has to go through inflammation. So inflammation, it's one key event. And like that, you have different things that have to follow before you have inflammation. And yeah, now how can I try to link all this to omics or proteomics mostly? So here, these are the omics techniques. So we also have genomic, but genomic, it's too static for toxicology. So yeah, maybe methylation of DNA could be important, but altogether, well, no one pays so much attention to that. Transcriptomics, yes, it's coming. It's coming forward, I could say like that. But actually, it's far from the phenotype. And proteomics, it's closer to the phenotype, but it's not so well standardized. And that's the difference here, that we need a way to standardize proteomics so that we can use it, at least for these purposes, for regulation. Metabolomics, it's also closer to the phenotype, but to standardize metabolomics, it's even a step forward. It's even more difficult. So that's the reason why I think that proteomics has such a future in risk assessment, in this developing these test guidelines. And the first thing we did was to try to identify which were the projects that had done already something with nanomaterials from the omics approach, using transcriptomics, but mostly proteomics. And there are some of these projects that dealt with that, but you can see they are not too many. There are these 3, maybe 1 or 2 more, 10-year-old projects. Some of them are just finishing. And yeah, we have some 5 data sets, no more than that. And that's not enough for us to look for, this is a mode of action of the mechanism. This is not enough to figure out what is happening. But we also ask ourselves, do we expect such a unique mode of action for nanomaterials? Can it be that they have a toxicology that it's so unique that it's not represented by anything else in the world? And no, we expect that some other substance can also react or can have an effect like the nanomaterials. And that's why the first approach was, well, we don't have so much of nanomaterials. Let's go to see what is known for other lung alterations. And we went to the PRIDE. The PRIDE, it's one repository of proteomics data. And yeah, there are 16,000 projects. And we looked for everything that is related to lung. And well, here the slide is not complete, it's a little bit missing, but we collected everything that it's, as you see, substances, which effect they have, can be in vivo, can be in vitro. We took everything, we just looked for lung and that we could see an effect, meaning no IPs or no phosphoproteomics. We just wanted to see just a full proteome experiment, and that's what we took. And you see that COVID, there are very many of them: influenza, smoke, pollution, diabetes, blood pressure, anything that you can imagine, it's there. And we collected all this information. And well, as you can imagine, all these projects are very different. The way they prepare the sample, it's very different. The different devices that they've been used for the results, they differ a lot. Some projects, they detect, I don't know, 200 proteins, some other detect 5,000. All of them, they are very different. And still, what our approach, and this is something I'm going to to show tomorrow. We developed a workflow to process each one of them so that we can get descriptors. So that, yes, of course, we pay attention to some things, but what we try to do is to collect everything and to process it in a systematic way that it's like kind of high throughput. Of course, 500, it's not a high throughput, but like that, like for what we are used, that we can process hundreds of projects in the same way. And for each of these projects, we try to bring descriptors, as you see, for all of them. And then we do the same for the nanomaterials. The nanomaterials, we have the projects that I mentioned before. We are also producing own data that we analyze the same way. And these descriptors, it's what we try to bind. The idea behind here, it's kind of, sorry, it sounds too bold, but when you watch Netflix, you watch a little bit, and then Netflix will suggest you things because of your taste. Then they have an algorithm to know if you like that, you will also like this other one, because most of the people behave like this. And that's what we aim at, that with the descriptors, if we have descriptors that have some coincidences in each of them, that then we can relate to the nanomaterials. Maybe we can know that some nanomaterials behave like, I don't know, like COVID. They cause such an inflammation, and then we have more information of COVID than the one we have for the nanomaterials. So maybe trying to bind the projects can bring us forward for what we want to see, for the effect of them. Yeah, so I would like to leave for tomorrow the workflow. I would like to show what it's that we do, and also which are the challenges that we have analyzing the data. This is mostly that sometimes you don't know what it's what. When you go to the repository, you have, this is file A, B, and C, and what its treatment, what its control, and how we can solve that problem. Or when you have outliers, how we can identify that, and so on. So all this, the lack of standardization, how we can still try to interpret the data and how we can use it. Yeah, that would be it. Thank you.
Thank you, Veronica, for this great, interesting presentation. It was great, really. Just to kick off the question part of your seminar, what is your opinion of the, thank you for bringing up the communication in science. Mm-hmm. What is your opinion of the communication of the pandemics?
Oh.
It was awful. Especially in vaccines, maybe?
So we worked, my institute, it's very close related to the RKI, and this is the institute that was he was in charge of the communication. And I see the way we have, how we have to talk. So we have to be very careful, and I imagine that for this institute it was the same at the beginning. So it's important that there is no panic. That it's a situation you always try to avoid. But it's also important to be fair and to bring the risk and to try to the people to be aware of what's going on. Yeah, communication, it's different. It's different, it's arguable. What I can tell you is that they followed the protocols that they have to follow for such cases. And I think from that point of view, it was impeccable. They tried to do it the way it has to be done. What works, it's all your opinion.
Thank you. Questions for Veronica? Yes, a lot of questions here. Lex.
Thanks, Veronica, for your presentation.
Welcome.
Really great what you're doing. I just one question about if they are you are considered to use the organ on a chip as a model for this, because this avoid to use animals, and then the translation to what happened in humans is more easy.
It's not what we do. We have another approach, and we tried with the proteomics. We have cell lines that we use. We are trying the co-culture, and there are many other groups that are experts in organ on a chip, and they put the efforts there. But yes, it's a lot being done in that direction too. Yeah.
Thank you.
Okay. Thanks a lot, Vero, for this very nice talk. I would have a conceptual question about this endpoint, what you mentioned, right? That when you do this in vivo, you always have the endpoint, because basically you don't know the mechanism and you don't care because it's in vivo, so you just look at the endpoint. And if I understood it correctly, when we go to in vitro, in order to be able to define these key events, we have to know the mechanism, right? But also for the in vitro assay, you would have to define an endpoint then, no? I mean that you say, okay, I go until point X, and that is then my in vitro endpoint, no?
Yes, yes, that's totally right. When we do, for example, cytotoxicity, you have the cells and you try to approach which are the doses that are correct. And then you know, okay, I measure how many cells are living. Then you make a graph and you know what you're looking for.
Yeah.
Yes, yes. Then you have this kind of endpoints where you need just a result. But yeah, for the toxicity, when you are in an in vivo setting, you just want to see if there is the pathology you are looking for. But to replace that, and this is not just me that I would like to stop using animals, this is the way that the world is going to, this is the direction. We have to know the mechanism, we have to learn of how the toxicity is done at the molecular level, but also at the organ level, and but always trying to understand why things happen.
And I have a second question. So you nicely showed how strict this is regulated with your examples from the rats and from the rat, you know, for the testing. So now my question would be, as one example, you know, you have this glyphosate study where people say it causes cancer, and then other people say it does not cause cancer. But then it's so tightly regulated. So how come, if something is so tightly regulated, so you know how many rats, how long the treatment, blah, 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 and then they have the opposite opinion?
Yes, so glyphosate, it's a hot topic. We can come to this, how to communicate, and that's why I'm not going to talk about glyphosate. But the thing is, let's say another substance, you produce the substance or you import the substance and you are the company. And you have the guidelines. And it's important, the justification, why you do that test or why you don't do that test. So each company will try to avoid as much as possible doing what they have to do, and they will always present a reason why they don't do that. And they will say, okay, and also we have to keep in mind that Europe as well as the USA, they try that these animal tests are not done for nuts, that they are done only if they are needed. So these companies, normally what they do is say, to avoid animal testing, we can use this paper from '82 where they saw that. And glyphosate or substance X, it's was not tested, but you see this one, it's almost the same. It's just one atom difference. And then they present that as a proof it is not cancerogenic. But then there is some other paper saying, yeah, they do not behave exactly the same. And that goes also to the dossier for the substance evaluation. So you have a company presenting results, scientific results, and arguing why these results should be accepted. And then you have people like my institute, or yeah, we try to find that, to bring that up and say, yeah, but that's not the same. Yes, here you use the software, but actually there's no reason for that. So there's always this concern, and it's not yet proven because the experiments were not done exactly following the guidelines. But there has been a justification why they presented another results, and it's always, yeah.
Keeping in mind what you show us in the transcriptomic approach with the guard signature, do you think that if you find a proteomic signature, you could use an RPA to standardize the measurements? I mean, to measure the signature just an RPPA instead of mass spectrometry, for example?
I don't really think we will find a signature at the protein level the same way.
Okay.
Because what we do now, we just take everything that is there, and I hope that in the future it will be better. And even if we make own experiments, yeah, that we will have more comparable things, but you don't always detect the same proteins. And it depends a lot of the settings, which cell line you have, and how it worked. That's why I don't believe that we will find a signature of proteins. And our approach, it's more related to how we can describe that with descriptors and not with the identity of proteins.
Okay.
I don't really think we will identify something that is always there. I don't know, ribosomal proteins maybe, but otherwise, I think it's more at the descriptor level. Pathways can be reactome or GO term, something like that, than proteins. Yeah.
Okay, thank you.
Thank you. We don't have much time, but Veronica is going to stay for lunch, and she's going to give another lecture tomorrow, so please free to approach her for more questions. Thank you, Veronica.
Thank you.